everyone, it's me Shanty and you're watching Biology Nowadays. In this lecture, we will see what phytohormones are, their main functions and characteristics, and also the biosynthetic precursors of different classes of phytohormones. And finally, we will have a short look on the synthetic plant growth regulators. From a single cell zygote begins the life of a plant. It does not simply increase its mass and volume as it grows, but it differentiates, develops, and takes shape, forming a variety of cells, tissues, and organs. This beautiful time-lapse video shows the germination of a pea seed. Watch the full video by clicking the link given in the description box below. Plants cannot move around like animals. So, the plant growth depends on the interaction of a number of external and internal factors. Examples for external factors are light, temperature, day length, and gravity. The main internal factors are chemicals called plant hormones or phytohormones. For many years, it was considered that there were only 5 groups of plant hormones: Auxins, Cytokinins, Gibberellins, Ethylene, and Abscisic acid. They are still the classical or traditional phytohormones, but during the last 3 or 4 decades, some more groups were added to this list. They are Brassinosteroids, Jasmonates, Salicylic acid, Strigolactones, Peptide hormones, and finally the phytohormone like Polyamines. We will see each of them in detail. First of all, auxins. They are indole compounds. Indole 3-acetic acid or IAA is the most common auxin. Auxins are produced in the shoot and root apical meristems or tips of a plant. They are also found in the immature leaves and seeds. Auxins promote stem elongation. They also promote apical dominance. Apical dominance occurs when the shoot apex inhibits the growth of lateral buds so that the plant may grow vertically. This kind of growth will help the plant to get more light to conduct photosynthesis. In this plant, there is an active apical bud, and it is in this bud the auxins are produced. Even though auxins promote stem elongation, they inhibit the growth of the lateral buds. As a result, the lateral buds will not grow into side shoots. Now, if I remove the apical bud, the auxin concentration lowers, and because of this, the lateral buds will grow into side shoots. Auxins promote the activity of vascular cambium. Vascular cambium produces secondary xylem as well as phloem and result in the thickening of stem. Auxins help in root initiation. They also help in fruit growth. As I explained in the case of apical dominance, auxins inhibit lateral bud growth. They also inhibit the abscission, or in other words, dropping of leaves and fruits. Now have a look at these Arabidopsis plants. Arabidopsis plants are used in various plant research studies. Here is a normal Arabidopsis plant with normal levels of auxin. But this genetically modified plant cannot produce auxin. See the difference in growth shown by these two plants. So from this, we can conclude that auxins are promoters of plant growth. Now let's see what are the functions of cytokinins. They are adenine derivatives. Creatine is the most abundant cytokinin. Cytokinins are produced mainly in root tips. Small amounts of cytokinins are also produced in actively growing plant parts such as developing shoot buds, young fruits, etc. Cytokinins are called so because they promote cytokinesis, or in other words, cell division. They also promote cell differentiation, that is, specialization of cells. They play a major role in promoting shoot growth. Cytokinins also promote opening of stomata and chloroplast development, thereby promote photosynthesis rate. They also cause movement of nutrients to tissues where their concentration is higher. They inhibit apical dominance caused by auxin, which means that they help the lateral buds grow into side shoots. They delay leaf senescence. Leaf senescence means aging and gradual death of leaves. Here, these two tobacco plants were grown under drought conditions for the same period of time. The plant that has less number of green leaves is a normal plant. There is no surprise that its leaves have become dry because that's how a plant reacts to a drought condition. The plant with lots of healthy green leaves is a genetically modified plant. The specialty of this plant is that during drought stress, it can produce more cytokinins, and since cytokinins reduce aging of leaves, the leaves are greener and healthier than the normal plant. However, cytokinins inhibit root growth. But overall, cytokinins are promoters of plant growth. Now, what about gibberellins? They are terpenoids. Gibberellic acid 3 or Ga3 is the best known. Gibberellins got their name because they were first isolated from a fungus, Gibberella fugicuroi. Gibberellins are produced mainly in developing seeds, also in shoot and root tips and young leaves. One of the main functions of gibberellins is that they promote seed germination. They help in mobilization of food stored in the endosperm to the growing embryo. Another function is promoting internodal elongation. Internode means the part of the stem between two nodes. Can you identify these tall plants in this picture? Then be ready for a shock. They are cabbage plants. Yes, cabbage plants treated with gibberellins. These two cabbage plants are the normal ones. In the gibberellin treated cabbage plants, you can see the internodal elongation caused by gibberellins compared to the normal cabbage plants. Gibberellins promote flower formation and promote the production of male flowers in the place of female flowers in plants like cucurbits. They also promote pollen tube growth and fruit development. Gibberellins inhibit seed dormancy. Overall, gibberellins are also promoters of plant growth. Next phytohormone is ethylene, which is the only gaseous hormone. Of all the phytohormones, ethylene has the simplest chemical structure. It is produced during seed germination, ripening of fruits, abscission of leaves, and senescence of flowers. Ethylene promotes aging and senescence of plant organs. It causes leaf epinasty or drooping of leaves. In this picture, among these two tomato plants, the one on the right side was exposed to ethylene. After some time, the leaves showed epinasty or drooping down of leaves. Ethylene also promotes fruit ripening. One of the main functions of ethylene is promoting abscission of leaves, flowers, and fruits. Ethylene is the primary and most important chemical involved in the process of abscission. It inhibits stem elongation. Overall, ethylene is a growth inhibitor. Next phytohormone is abscisic acid or ABA. They are carotenoid derivatives produced in the mature leaves, dormant buds and seeds. The name abscisic acid was given because researchers at first believed that it was this hormone that stimulated abscission or falling down of plant parts. Today, this name abscisic acid is a little misleading because it is now proven that even though abscisic acid promotes abscission, it does not play a primary role. As I said earlier, It is ethylene which plays the primary role in abscission. Abscisic acid promotes dormancy in seeds and buds. It also promotes closure of stomata under drought conditions so that less water is lost through stomata and helps the plant survive in such unfavorable conditions. In this picture, these two plants were under drought stress for the same period of time. Among these two plants, the plant treated with more abscisic acid analog shows better tolerance towards drought. As I mentioned earlier, together with ethylene, ABA promotes abscission of leaves. Abscisic acid is a general inhibitor of plant growth. It also inhibits seed germination. So overall, abscisic acid is an inhibitor of plant growth. Even though when I say that ethylene and ABA are growth inhibitors, under certain unfavorable conditions like drought stress, it is necessary to slow down or inhibit the plant growth to make sure that the plant uses less water and survive the drought conditions. Because under drought, the strategy is to survive rather than grow. So, growth inhibitors are also actually good and necessary for the plant growth and survival. The sixth class of hormones is the Brassinosteroids. They are steroid compounds. The most abundant among the Brassinosteroids is the brassinolide. It was first isolated from the pollen of Brassica napus, the rape plant from which we get the canola oil. Brassinosteroids are found mainly in pollen and immature seeds. They promote cell enlargement and cell elongation, cell division, pollen tube growth, seed germination, xylem differentiation, which means specialization of xylem cell types, and resistance to stress. Among these two cucumber plants, the small plant is a genetically modified plant which produces less brassinosteroids than the normal plant. From the difference in growth exhibited by these two plants, I hope you can understand how important brassinosteroids are for the normal plant growth and development. So, brassinosteroids are promoters of plant growth. Next group is the Jasmonates. They are lipid-derived cyclopentanone compounds. A member of this group is methyl jasmonate. The isolation of methyl jasmonate or methyl ester of jasmonic acid from jasmine oil derived from Jasminum grandiflorum led to the discovery of the molecular structure of jasmonates and so their name. The scent of jasmine mainly comes from methyl jasmonate. Jasmonates are produced by plants under stress. Jasmonates promote initiation of defense reactions against pathogen. In a recent collaborative study conducted at Utrecht University and University of Amsterdam in the Netherlands showed that when two Arabidopsis plants infected with a pathogenic fungus, the plant with more jasmonic acid exhibited less disease symptoms. However, it showed less growth than the normal plant. This is because of the growth inhibitory effects of jasmonates. Jasmonates promote senescence of leaves. They inhibit seed germination and root growth. Next is salicylic acid. It is a phenolic acid. It was first isolated from the bark of willow tree. The word salicylic acid was derived from the Latin word salix, which means willow tree. Salicylic acid promotes resistance to pathogen by inducing the production of pathogenesis-related proteins, that is, proteins produced in plants during a pathogen attack. It promotes protection to various environmental stresses. For example, these two plants were grown at a temperature of 40 degrees Celsius. The plant on the right side was treated with salicylic acid, and this plant showed better tolerance by growing better than the untreated plant. acid promotes the levels of chlorophyll and carotenoid pigments and as a result promotes photosynthesis. It also promotes flowering. Now the next group of phytohormones, the strigolactones. They are terpenoid lactones. Strigol was the first isolated strigolactone. Strigolactones are produced in roots. They promote seed germination of parasitic weed, for example striga. And because of this role, these phytohormones got their name. They also promote recognition of the plant by arbuscular mycorrhizal fungi and promote branching in the arbuscular mycorrhizal fungi. They promote plant height and secondary growth. Strigolactones promote the formation of root hairs. They also promote senescence of plant organs. They inhibit branching. They also inhibit lateral or side root formation. Next group is the peptide hormones. They regulate cell division, reproduction, nodulation, senescence, defense and environmental responses in plants. Some examples for peptide hormones are cysteamines, phytosulfokines, CLV peptides and PLS peptides. The last group is the phytohormone like polyamines. They play a role in the modulation of senescence of plant organs and in the regulation of programmed cell death in plants. Examples for polyamines are putrescine, spermidine and spermine. Now let's see the biosynthetic precursors of the main phytohormones. Even though the phytohormones have diverse chemical structures, most of them are derived from three main types of metabolic precursors: amino acids, isoprenoid compounds and lipids. The amino acid tryptophan is the precursor for auxins. Methionine serves as precursor for ethylene and phenylalanine is the precursor for salicylic acid. The isoprenoid pathway gives rise to 5 different classes of plant hormones. Here, isopentenyl pyrophosphate or IPP serves as the main precursor. Cytokinins, Gibberellins, Abscisic acid, Brassinosteroids and Strigolactones are the 5 classes of phytohormones synthesized through isoprenoid pathway. Jasmonates are synthesized from a lipid precursor, which is alpha-linolenic acid. Now I hope that you got a clear picture on the functions and biosynthetic precursors of different classes of phytohormones. Actually, the word hormone comes from Greek word hormone, which means to excite or to stimulate. Because at first people thought that hormones always stimulate or promote growth. But now we know that hormones can promote as well as inhibit growth. Let's see some of the characteristics of phytohormones. They are naturally produced in plants. They are present in extremely small quantities. For example, in a pineapple plant, there will be only 6 microgram of indole 3-acetic acid per kilogram of plant material. I know it's very difficult to have a feel of such a minor quantity. So let me put it in another way. It is like a big Volvo bus of 20 metric tons carrying a small needle in it. That's interesting, right? The third feature of phytohormones is that the site of synthesis and the site of their action can be different. For example, as we saw earlier, cytokinins are mainly produced in the root tips. But besides affecting root growth, these cytokinins are transported to shoot where they affect shoot growth also. Next characteristic of phytohormones is that they often interact or do crosstalk with each other. Let me explain what this means. This is a summary of what we discussed about the main functions of the 5 classical hormones: auxins, cytokinins, gibberellins, abscisic acid and ethylene in the 6 main plant processes: seed germination, general growth, flowering, fruit development, abscission, and seed dormancy. Now consider the case of growth. Here auxins, cytokinins and gibberellins together help to bring about the effect of growth in plants. Such kind of acting together to promote each other's activity like friends is called synergetic interaction. Now let's take the case of seed germination and seed dormancy. Here gibberellins promote seed germination and inhibit seed dormancy, while abscisic acid inhibits seed germination and promotes seed dormancy. So such an interaction where 2 phytohormones have opposite effects on same process is called antagonistic interaction. Finally, let's have a short look on the synthetic plant growth regulators. Firstly, synthetic auxins. As the name indicates, they exhibit auxin or growth promoting activity. For example, 2,4-dichlorophenoxyacetic acid. It is commonly called as 2,4-D, and indole 3-butyric acid or IBA. There are some synthetic plant growth regulators that come under the category of growth retardants or growth inhibitors. First group is morphactins, meaning morphologically active substances. They have an inhibitory effect on plant growth. Then antigibberellins. They act by blocking the main steps in gibberellin biosynthesis in plants. Examples for antigibberellins are phosphon D, cycocel, and ammo-1618. The next synthetic growth retardant is malic hydroside, which acts by inhibition of cell division. And that's all for this video. Please help us to improve the quality of this channel by giving your valuable suggestions in the comment section below. Thank you for being with me and stay tuned.